Second step is a platelet release reaction or secretion. Now, after platelets are adhered to the collagen through a 1 Willebrand factor, something interesting happens. Inside the platelets, there are special secretory granules which release substances. And this process is called a platelet release reaction. There are two types of granules. First is a delta granules. Second is a alpha granules. Let's talk about delta granules first. These delta granules are very dense, so they are called dense granules. This dense granule release some substances. To remember which substance, look at the picture that has been shown. Delta granules or dense granules look like a sag. So remember this word sag. S stands for serotonin or 5-hydroxytryptamine. A stands for ADP, that is the adenosine diphosphate. C stands for calcium ion. Now, what are the function of this substance? Serotonin. It causes the vasoconstriction. And yes, we need the vasoconstriction at the injured site, right? ADP or adenosine diphosphate. It is a powerful platelet aggregator. You will ask, what do you mean by platelet aggregator? Platelets are very sensitive to the ADP. That means as soon as ADP molecule touches the surface of platelets, platelets become activated, change its shape, and platelets are start to stick each other and form a clump or plug at the injured site. Platelet aggregation means platelets sticking together to form a plug. Here's the beauty. Some activated platelets release ADP, which cause more platelets to get activated and release even more ADP, amplifying this whole process. This results in the formation of a plug or clump known as a primary platelet plug or primary hemostatic plug, or also called a temporary hemostatic plug, because this primary aggregation of a platelet is reversible and it is a loose platelet plug. Calcium ions. Calcium ions are released from delta granules. They have an important role in the coagulation process. Calcium helps to hold the coagulation factor tightly on the phospholipid layer of a platelets so that coagulation process occur effectively. Now, second is alpha granules. Alpha granules release fibrinogen, fibronectin, and platelet-derived growth factor. Here, fibrinogen is one to focus on. The word fibrinogen comes from the word fibrin generator because it is the precursor of a fibrin. Fibrinogen is converted into fibrin and this fibrin gets deposited over the primary platelet plug, forming a strong mesh around it. This transforms the primary platelet plug into a strong, stable plug. Now it is called a secondary platelet plug. Now, let's go back to the something interesting. During the platelet adhesion, when platelets stick to the collagen via 1 Willebrand factor, this triggers the activation inside the platelets. So, inside the activated platelets, an enzyme called phospholipase A2 gets turned on. This enzyme breaks down the platelet membrane phospholipid into a fatty acid called arachidonic acid. This arachidonic acid then converted into the thromboxane A2. Thromboxane A2 is released from platelets into the surrounding area and it causes the vasoconstriction. Thromboxane A2 has a similar effect just like ADP. So both thromboxane A2 and ADP promotes the platelet aggregation, meaning platelets sticking to each other. Now, here's the something really cool. Healthy endothelial cells secrete prostacyclin, which is a vasodilator and inhibits the platelet aggregation. On the other hand, thromboxane A2, a platelet-derived prostaglandin, is a vasoconstrictor and promotes the platelet aggregation. There is a constant competition between prostacyclin and thromboxane A2, and interplay between them defines the balanced regulation of a platelet function. For instance, in a normal state, prostacyclin is a more dominant and it prevents the intravascular platelet aggregation. But after injury, thromboxane A2 becomes more dominant and it favors the platelet aggregation.